So I'm going to start by saying that this is really a talk which has a lot of biology, and so it's a general talk. I'm going to assume you know very little biology. I'm going to spend a lot of the time introducing you to new words and new concepts, and maybe you'll find there's very little physics in there. But one of the messages I want to convey is that there is a lot that physics can contribute to biology. There's a lot that it has contributed. And I would go even further and say that without the contributions from physicists, biology will not be as rich as it deserves to be. So, please, all of you, try and look around and find examples where life sciences and your own work intersect. It's really a very, very exciting time to do such a thing. So the first slide of this talk is not really part of the talk. It's more motivational, and it has to do with my interests, particularly in living systems. The problem when as a physicist you get into biology is there's too much to study. Are you studying molecules or cells or brains or ecosystems or so at some point you have to decide which scale of biology, what objects you're interested in. So I'm trying to motivate that, right? And if you wanted to pick a big motivating question, this would be a nice one. I'm sure all of you have thought about it. What would life look like on other planets? The fact that you can ask this question as a scientific question is remarkable. It means that we somehow have a definition of life and understanding of what makes it a general definition, not just life on Earth, and we're trying to extrapolate it. And I think it's a very good thing to think about as a thought experiment. One piece of what we would expect life to have on any planet is evolution. And the reason is almost definitional. We can't imagine any other process that leads to the increase in the complexity of objects that are able to mobilize resources from their environment in order to, for example, propagate themselves. Darwinian evolution is the one mechanism we know that does that. So in some very generic sense, life and evolution cannot be decoupled. Fine. This is more interesting. Two of the great strands of 20th century science. One involves the development of information theory and the idea of the digital computer, and the other one involves the discovery of DNA as the substrate for inheritance, and the fact that DNA is made up of these 4 bases, ATGC, and looks almost digital as a digital storage platform. It's not accidental, because errors can be corrected very, very reliably in a digital substrate, and this is the reason why heredity goes through DNA, among many other aspects of what a cell gives to its offspring. So information as a general principle that unifies how biological systems are organized, I think is a very good thing to unify life on other planets to life on Earth. What about this one though? Cells. So cells, you'll hear a lot about cells in my talk. What is the definition of a cell? I'm not talking about a cell that has lipids on the outside, that's exactly 10 microns in size, that has a nucleus. Not like that. A cell, broadly is a thing that has an inside and an outside. That's all. And in some sense, that boundary is essential for information and evolution to work. That's because Darwinian evolution works when a mutation, if it produces a useful benefit, that benefit should be kept private and not shared publicly. If it's shared with everybody, then the strength of evolutionary competition doesn't work. If it's kept private, then it does work. Okay. So in some sense, the boundary, which is what a cell provides, is a very important ingredient for Darwinian evolution to function, at least effectively. And in this sense, I think cells as a concept are absolutely universal to the definition of life. I work on cells. This is part of the reason for that. Okay. That's just sort of broad motivation. This is the rest of my talk. It is a talk about cells, and it's a talk about what we call complex cells. I'll explain what I mean by that. And it's about very, very recent discoveries that are being made about the organization of all living things on Earth and things that unify them all, patterns of life. So I'm going to start with a lot of history, and bear with me, because even if you think you've read about this in the textbooks, you'll find that what you know maybe is out of date. So here's something which by now is well established, that all life, whether it's microscopic, invisible to the naked eye, or macroscopic, blue whales and trees and human beings, all life is made up of cells. This wasn't always known, but it is a fact, completely well established now. And it really got started with the idea of trying to find the microscopic organization of life after the advent of the optical microscope, right? So this is Van Leeuwenhoek, this is Robert Hooke, both contemporaries, both had microscopes, very good ones. They were both very good observationists. So Van Leeuwenhoek actually looked in samples he picked up from the environment, like just ditch water, and he'd put it under the microscope. This is an example of that. And he would see in this little ditch water an entire ecosystem of predators and prey and photosynthetic organisms and colonies. And these look very much like ecosystems of large animals. And so he called them animalcules. And we know now that these are cells. You can actually take the cells that he drew in his notebooks and you can make a one-to-one map to electron micrographs from data that's collected today. He was just beautiful. The level of detail that he captured in his observations is just stunning. At the same time, Robert Hooke was looking at macroscopic things, macro, big trees, plant tissue. And when he sliced the plant tissue, put it under the microscope, he found that it was broken up into repeating units. And these repeating units he named cells. He named them cells because they look like the cells of a monastery. That's where the word cell comes from. And very quickly after this, it was realized that these units are actually the same as these at a subcellular scale. And it's this unit, either in multicellular form or unicellular form, that comprises all living things. So this is an astonishing idea and a very unifying idea. This means that, yes, the same hook. Okay, so once you've found this unifying aspect to life, all life is made of cells, you again want to break it up, right? So one of the motivations of science is phenomenological. You want to classify things. Although all things are of one sort, what are the subsorts? How are they closer related or more distantly related? That's the impetus to classify. So in Hooke's time, it would have been that there are two types of living things, macroscopic and microscopic. And it seems that all the macroscopic things are related to each other, and somehow all the microscopic things are related to each other, and these are two different things. This is an obvious way to look at things. It turns out to be wrong. And the real difference between different types of cells is actually something very technical and not exactly visible to the naked eye. And it has to do with these words, prokaryotic and eukaryotic. I'm flashing this in a physics audience, and I hope it doesn't put you to sleep instantly. But I will tell you something very surprising about this. The words prokaryote and eukaryote, when did they enter the textbooks in biology? Just quickly try and guess when that is, and I'll tell you the answer in about 3 seconds. Okay, in the 1970s. Not before that. Okay, so this is a very, very recent idea that the classification of life is along this particular axis. And again, the reason for this is it's not visible to the naked eye. It had to wait for the application of electron microscopy to look at the well, at least the molecular organization or the subcellular organization of cells. Once the electron microscope was used to examine cells, it became clear that there were two types of cells, but they were not the macroscopic guys and the microscopic guys. It was something called prokaryotes and eukaryotes. And this is really an amazing quote from a paper in 1961, this paper by Stanion Van Niel. You would think that by this time the idea of what a bacterium is well established. You all know what a bacterium is. There was no definition of a bacterium in 1960. And in 1961, here's the definition they came up with. Okay, so this is interesting. Since the earliest days of microbiology, the biological nature and relationships of the bacteria have been subjects of discussion. Any good biologist finds it intellectually distressing to devote his life to the study of a group that cannot be readily and satisfactorily defined in biological terms. There was no definition of a bacterium in 1961. People knew a lot of bacteria, but what was the common feature of all of them? And they came up with the definition. Here was the definition. They said that there are these other type of cells, which are called the eukaryotes, meaning that they have a nucleus, among other things. Okay, eukaryotes are called eukaryotes because they have a nucleus, but actually they have a whole bunch of other features that unify them, and I'll go over that in the rest of the talk. So this is from a paper in 2005 talking about the 61 paper. Stanley and Van Niel still could do little more than define the prokaryotes, the bacteria, in negative terms in relation to the eukaryotes. Eukaryotes, which are the kinds of cells that make up us, or trees, or amoeba, or paramecium, unicellular and multicellular, everything except the bacteria, as far as people knew in 1961, had a membrane-bound nucleus, a cytoskeleton, which all these things that push and apply forces, intricate system of internal membranes, mitochondria, chloroplasts, and so on. And what is the definition of bacteria? They are the anti-eukaryote. Bacteria, smaller, and lacked all of these structures. They lacked mitosis. So this is astonishing that there was no definition of a bacterium in 1961. And then when the definition came, it was somehow that the eukaryote was the primary thing and the prokaryotes were the secondary thing by definition. So this is perplexing because really you would like to start from something simpler and define up to more complex forms, right? That had to wait another decade. Carl Woese embarked in the late 60s on what turned out to be a very modern way to classify biological systems. He said, instead of looking at it under a microscope and using your arbitrary subjective criteria to say this is one group, this is another group, this is a third group, we're going to define it mathematically. Because by then, the sequencing of DNA and RNA became widespread. You were able to do it reliably. And DNA sequences, it's hard data. You can come up with quantitative metrics. Are these two sequences close? Are these two sequences far? And in that way, you classify two organisms as being close or far. What he did was he looked at a gene called ribosomal RNA, which is conserved across all organisms. He sequenced them. He then ran the sequences through an analysis pipeline that produced what's called a phylogenetic tree. This is not Wose's tree, but it's something very much like he would have got. A phylogenetic tree at one level is just a quantitative map saying that these are all living species at the leaves, and two species that branch more closely are more closely related quantitatively because their DNA sequences are similar. Phylogenetic trees, as Darwin realized, are also representations of how things evolved over time, which means that it not only tells you the relationship between present day organisms, it tells you that there was an ancestral organism whose descendants diverged and became several different species that we see today. Okay, that's what a phylogenetic tree is if you're not familiar. This is the tree that Carl Woese found, and he was expecting, based on what people knew from the 60s, that there were two groups, the eukaryotes, which he did find, and the prokaryotes. What was astonishing, and it just dropped out and nobody expected this, is that the prokaryotes are not a single group. They're actually two completely different organisms. So different that, they could have, you could say they could have come from different planets. Right? They are called the bacteria and the archaea, and prior to Woese, people assumed that these were all the same type of organism, but they're not. They're completely different at the molecular level. The lipid chirality is different, the translation mechanism is different, DNA replication is different. Basically the genetic code is the same, but everything else is different. So from Carl Woese onwards, the classification of life at the highest scales has been, you have the eukaryotes like us and trees and blue whales and paramecium. Then you have the prokaryotes broken into two groups, the bacteria and the archaea. And I'm spending so much time on this slide because this classification has been the correct one. In those 50 years or 60 years since it was developed, it hasn't been overturned. Okay, so this is at one level the answer to the question. But then the next question comes. Life has to evolve from one form to another. So if there are three groups, which one was first, or was there some other group that was first from which these three evolved? What was the history of them, not just the present day classification? That becomes the next obvious question. And as far as Woese and others were concerned, this is, these trees, technically you cannot root them. You don't know where they start in time because mathematically it's all the same. But what they had concluded was that the root of this tree is right there, and therefore the bacteria form an outgroup. The eukaryotes and the archaea are related to each other. They're kind of cousins. That was sort of an assumption, but it was not very well established because there's a big error bar at the center of this tree. Okay. So flashing forward to today, here are some very recent papers that capture the diversity of all life on Earth. Everything you've ever seen or heard of has been in a lab. All those cells are represented here. I've shown it as two different trees. One, okay, I'm sorry for the resolution. You can't see much of it, but one is the bacteria, which are these blue guys and that orange group, and the archaea, which are these purple and pink guys, the prokaryotes. They're certainly related to each other, but quite separated by a long branch. That's the tree of prokaryotes. And here you have the tree of eukaryotes, like us. But what I want to point out over here is that among the eukaryotes, all animals, all fungi, all yeast, okay, live inside that little branch. Okay? And inside this little branch live all the plants and the trees and the rice, wheat, everything else in this little branch. Animals, fungi, and plants occupy these two branches. And if you learned that that's what life is in school, it's obviously totally wrong, because all these other forms of life dominate the diversity of eukaryotic life. It's taken a long time to get this tree because in some of these lineages, we have only one species example. People go into a deep sea vent somewhere and they find some new type of life and they have to add it to the tree and hope it falls into the existing group. Okay, so that's one point I want to make from this tree. So you would have heard of some of these actually. I mean, so for example, these are ciliates, these are diatoms, these are single-celled algae, these are trypanosomes which cause disease. So you would have heard about all of these, right? But the fact that we can now classify them is a new thing. And I think the classification of eukaryotes is something which wouldn't have worked even 10 years ago because we didn't have enough genomes of different organisms. So everything I'm telling you is actually quite modern. Now the final question. You have the prokaryotes, you have the eukaryotes. How are they related to each other? What's the idea? So in the next slide, this is called spoiler alert. I'm just giving you the end of the whole talk. Eukaryotes, where do they come from? They don't come from bacteria. They don't come from archaea. They are a mix of both. Okay, and this should surprise you no end, because if you read Darwin, or if you understood evolutionary biology, you would know that life arises and diversifies in a tree-like manner. It's not that things come together, it's that things move apart. Okay, but the story of the creation of us actually involves something called hybridization, and I'm going to explain to you how this works. What is an so they live on your teeth. They're single-celled organisms that live everywhere. Many are extremophiles. They're different from bacteria, but they look like bacteria. That's why until you sequence them, you couldn't tell the difference. Yeah. So you do have archaea living on your teeth. The reason, by the way, that archaea are not well studied is that they do not cause human disease. So we don't care about them. I mean, that's a joke, but that's the reason why they've not been studied. Okay. So I've told you that eukaryotes are a hybrid of bacteria and archaea, and I'm going to explain to you what this means. But who came up with this idea? It was floating around for many years, but the person who really pushed it over the finish line was this person, Lynn Margulis. So in the 1970s, she argued very forcefully, backed up by data, cell biological, electron microscopic, and so on, that the mitochondria, which all of you know from textbooks, they live inside you, they're inside your cells, they are the site of oxidative respiration inside your cells. Those things used to be free living bacteria. They used to live on their own and somehow ended up inside another cell. And she had lots of lines of evidence. The membrane of the mitochondria is analogous, homologous to the membrane of bacteria. And as I'll show you in the next slide, mitochondria have their own DNA, which you might have heard about. So lots of lines of evidence. I really like this slide because this is Lynn Margulis' wedding day to Carl Sagan. And it was during her marriage to Sagan that she actually published a series of papers about mitochondrial endosymbiosis. Everybody knows Carl Sagan, or you guys are too young? Okay, okay, just checking. So mitochondria have their own DNA. So this is, any eukaryotic cell, the blue stuff is the nuclear DNA and the red stuff is the mitochondrial DNA. And this is one line of evidence that they used to be their own creature. Okay, there are many, many other lines of evidence. Fine. So if mitochondria were bacteria, what was the host? What was the host cell, right? It's another big question. Okay. So here you need to start looking at the timeline and trying to see whether you can pull together different lines of evidence. Very recently, and these are two very recent papers from 2023, which I encourage you to read, people have started to date the origin of eukaryotes using fossil evidence, chemical evidence in rocks, and so on. One timeline, which I forgot to mention here, but I should. So mitochondria do oxidative respiration, and there was no oxygen in the Earth's atmosphere prior to 2.5 billion years ago. Because prior to that, there was no photosynthesis. Photosynthesis was invented by bacteria 2.5 billion years ago. They're called cyanobacteria, which led to, over time, the accumulation of oxygen in the atmosphere, after which photosynthesis could happen, respiration could happen, and therefore eukaryotes must be at least younger than 2.5 billion years ago. Okay, so here's a timeline of billions of years ago. Here's 1 billion years, 1.6 billion years. What you find in rocks, in sort of rock strata in different locations, is that the early rock strata tend to contain prokaryote-like chemicals of various types, which is in pink here, and the shallower ones, the more recent ones, contain eukaryote-type chemicals, and you can actually date these, right? So you can put a bound on at least how old eukaryotes have to be based on this. At the same time, there are fossils of cells. You can actually find micron-scale fossils of cells in the rocks. It's a very difficult job, but you can do it. And you do find that after this about 1.6 billion year time scale, there are fossils of eukaryotes that show up in the records. I'll show you more beautiful pictures of such fossils. But based on this, we know now that prokaryotes are old. In fact, they're about 3.5 billion years old, and eukaryotes are younger. They're about 2 billion years old. So the story is that there were prokaryotes, and from there eukaryotes came. Okay. The other piece of evidence, which has always surprised me, and again, it's very, very unexpected. You can take the genes of humans or plants or blue whales or any eukaryotic cell. Every single gene, you can ask, how does it relate to the other two groups, meaning archaea and bacteria? Did that gene come from archaea or did it come from bacteria? You expect it comes from one or the other, depending on which one we came from. They come from both. Some of our genes actually come straight from bacteria, and some of our genes actually come from archaea. And more or less it's even numbers. And there's a pattern to it. the genes in our bodies that are responsible for all kinds of chemical synthesis, metabolism, the stuff that makes energy, those things come from bacteria. But the genes that are responsible for DNA replication and protein synthesis and so on, those come from archaea. There's a pattern to this, okay? So there's something very compelling about what's happening over here. It's telling us something. It's not clear exactly what. So let me just stop here and repeat that assertion I said earlier. In biology, the tendency is to assume diversity comes from competition and divergence. But here's an example where a new type of cell was created. In fact, it's the biggest leap in life on Earth since the origin of life by the coming together and the mutual cooperation of two different types of cells. And so the idea of cooperation as driving the creation of new biological form is actually quite common, but very poorly studied. And here's a stereotypical example of that. Okay, fine. So what was the host cell then? We know that mitochondria were bacteria. We know that there's an archaeal contribution. But can we find any examples of those cells that are still alive today? The ones that came together to make us. So with the mitochondria, we can, it turns out, that we know enough bacteria, and among all the bacteria, we know one group, which are called alpha proteobacteria. You know one famous example of that, it's called E. coli. Actually, is very much like the bacterium the mitochondria used to be before they became tame. So now you could ask the same question on the other side. We know that the closest living relative of mitochondria is alpha proteobacteria, like E. coli. What is the closest living relative of the archaeal cell from which we came? Can we find it? So until 2010, there was no answer to this question, but 2010 there was a major discovery. Sorry, I promised you that I would show you some beautiful electron micrographs of eukaryotes. So clearly by 1.6 billion years ago, there were cells that you can find in rock strata that are big. I think they're about several tens of microns, and they have a lot of internal structure, very much like present day cells. So clearly by 1.6 billion years ago, there were eukaryotes. Okay, so we have to explain where they came from. Okay, so here's the question. We know that eukaryotes are complex, they're big, they have this internal structure, and I know maybe many of you are asking this much along the talk, where's all the physics coming in? So my claim is that this physical architecture of the cell is an important lens through which to understand the evolutionary story. The physical architecture of eukaryotes is unique and very different from what we know about prokaryotes, at least what we knew about prokaryotes, say 20 years ago. So if we think that eukaryotes came from bacteria, which we know are very simple, and archaea, could it be that the complexity of eukaryotes is already there in the archaea? Somewhere there's some very complicated archaea which is in some sense related to the ancestor of us, and the addition of the mitochondria is just a decoration. Or is it that somehow a simple archaeon and a simple bacterium came together to make a eukaryote? Okay, these are two different hypotheses, and we didn't know anything about it, let's say 20 years ago, but recently there was recently meaning in 2010, there was a huge breakthrough in the story, and the answer comes from Asgard, and I don't mean from these guys, which I guess you all know, but I mean from here. So this is called Loki's castle, and it's a deep sea vent, and it's 2.3 kilometers below sea level off the coast of Norway, and there was an automated submarine expedition to dig up silt and bring it up. I mean, people do this kind of thing all the time. And the point about this expedition was to bring that stuff up and sequence what was in there. You can't grow what comes up from there very easily because they are designed, they adapted to grow at high pressure. So you have to take great care to actually grow them in the lab, but you can sequence the DNA because that will survive all the way to the top. So they sequenced the DNA of stuff that was in here, and what they found was in some sense a smoking gun. They found the sequence of presumably a cell, okay, a genome sequence which looked archaeal, so it was archaeal, but as close to eukaryotes as any archaea that's ever been found. So they found the close archaeal cousin of eukaryotes, and it was sitting there in the silt 2.3 kilometers below the surface of the sea. So here it is, it's called Loki archaeota, and in fact after that the same type of cell has been found not just in the deep sea but basically in muddy sediments at sea level and many different ecosystems, and now there are many examples of these. And because the original one was called Loki archaeota, they called the whole thing the Asgard archaea, and they named them Heimdall and Thor and all that, which is a nice bit of marketing, I think. So these are the Asgard archaea. From in there is where the eukaryotes came. From where, in fact, related to that particular group called the Horde archaea, right there, because the eukaryotes branch out from inside this lineage. Okay, plus the bacteria came somehow. So I know I'm throwing up a lot of different pieces of the story to you, and I'm going to try and bring it together in the next slide. So here's the question. That archaeon that we came from, was it already complex? Now, it turned out it's very difficult to culture these. It took a long time, but in 2020, it was finally cultured. You ask what archaea look like. Typically, they look like bacteria, but this one doesn't look like bacteria. It looks very, very different. It has these protrusions. It has an internal cytoskeleton, which is different from the eukaryotic one. This is not like that. Yeah, those are, rod-like and whatever, floppy. So these things, in some sense, they are atypical archaea. So they're not the same as all the rest. They have these protrusions, but in another sense, they're typical. They're very small. They're just a micron in size, and internally, they don't have too much structure. So the answer is that the archaea we came from was very simple, and the bacterium we came from was very simple, and somehow the complexity came later. This is what the complexity of the inside of a eukaryotic cell looks like, right? The Golgi apparatus and vacuoles and nucleus. Nothing like this is present in the prokaryotic domain. So somehow it came afterwards. So here's the story, right? And it's about halfway through the talk, which is where I should be. At this point, I've given you all the data that people have gathered. And now you're in the realm of hypothesis, and here's one of the more likely scenarios I'm presenting, but this is subject to change. So how does this happen? So it turns out that these archaea live in symbiosis with bacteria. They live with bacteria studied on their cell surface, and that's why they have all these protrusions to increase the area of contact between the membranes of these two cells, and these cells exchange food and they exchange gas. So they live in what's called a mutualistic relationship. Yeah. So you have a blue archaeon, you have these red bacteria, and in the present day, these kinds of archaea live like this in tight conjunction with bacteria. It doesn't take much of a leap of imagination to see that if these protrusions became big and fat and engulfed those bacteria, eventually, over time, you seal the outside. Suddenly, these things start to look like mitochondria. You automatically have a nucleus, which is the remnant of the original cell, and you have an outer membrane system, which is very, very complex. Okay, so this is a story. This is a story, but it's a nice story because it creates the mitochondria and the nucleus at the same time. It's still insufficient because it doesn't say where all the other complexity of the eukaryote comes from, which is the kind of thing that we study. So here we are today. So what do I study, which is what I'm going to spend the rest of the talk discussing. So I'm interested in the internal architecture of eukaryotes, specifically what are called organelles, which are these complex membrane-bounded compartments like the Golgi and the ER and the endosomes and the lysosome and so on, which I showed you in the EM. So I'm interested in where they came from, how they get set up in a cell, and how they change over time. So I'll show you, going from the broad landscape of our work to more and more specific things as the talk goes along. But here's the timeline again, okay? So if you believe that story, ancestrally there was an archaeal cell in tight coupling with some bacterial cells. And over time, this relationship got tighter and tighter until eventually the population contained cells that had basically the mitochondria living inside the other cell. And remember, very, very important, this doesn't happen that one cell of an archaeon got one cell of a mitochondrion and we came from there. That's not how it, there's a whole population, and over time the population becomes closer and closer in interactions, and over time you get a new type of organism coming out of that, which it could take a long time. So this took a long time. After the origin of what are called the proto-eukaryotes, that is the first cell that had mitochondria internally and a nucleus, over time there must have been a lot of diversification. We don't know where on earth they arose, but wherever it is, they survived, they diversified, just like humans arose in Africa. Then there were many human types, right? A lot of those human types that lived at one time on earth, like the Denisovans, the Neanderthals, and so on, they're all extinct now. And so we have to go much further back to find a common ancestor with the apes. In the same way, a lot of the eukaryotic diversity that was present in the early days became extinct. So what's left today are the descendants of a rather modern cell. And that rather modern cell is already astonishing in its complexity. It has mitochondria and nucleus, but it has this entire system of other membranes and cytoskeleton and mitosis and all this cool stuff you associate with eukaryotes. These features arose on sort of a billion-year branch. But because we don't have those intermediate forms, we can't tell in what order those traits arose over time. It's just a matter that we don't have those descendants of those collateral forms surviving to the present day. These cells then diversified over time to make that wonderful eukaryotic tree that I showed. But I should tell you, at the cellular level, it's amazing. Whether you take a plant cell or an animal cell or a protozoan cell, at the subcellular level, it's astonishing how conserved all eukaryotes are. So that cell and this cell will be very, very similar, and this cell is then given a name. It's called the last eukaryotic common ancestor. So one of the big projects in this field is what was that, what did that population of cells really look like? Okay, so we've worked at many levels over here. We worked at the, and I'll show you some snapshots of this in the next few slides. We've worked on the archaeal origins of eukaryotes. We've worked at the host mitochondrial integration. We've worked at the origins of this thing called phagocytosis, which is the process by which eukaryotes get food. And we've worked on the emergence of new organelles over evolutionary time, the last of which I'll spend a lot of time on. So here's an interesting point. Why did it take a hybridization to make eukaryotes? What is it that the eukaryote has that the two independent prokaryote groups did not? So if you're interested in membranes, imagine you're sitting in a cell, there are many different membranes. They're all made of lipids, right? Why do they look different? How can I send stuff to one membrane and not to another membrane? It's because they're all decorated with certain chemicals, and they're decorated with different, you can think of painting them different colors. So anything in a cell knows where to go because there's a marker that's telling it where to go. One of the main markers in a cell are something called small GTPases, okay, which is proteins, which are attached to membranes by lipid tails. Now here's a nice twist. Those small GTPases we inherited from archaea. But the lipid tail, the machine that makes that, we inherited from bacteria. And the two different cell types independently didn't have this mechanism. But when they came together, you had this new mechanism. It's one of the many things where at this hybrid level, you had the power to do new things which you didn't have before, which is very, very interesting. I mentioned this thing about mitochondria bring free living. You can imagine that a bacterial cell is not happy living inside another cell because its growth and division, it has its own motivations to grow and divide. A cell that grows and divides inside another cell too rapidly, we have a name for it. It's called a pathogen, right? It's an infection. It kills you. So if you want a symbiosis, the host cell and the symbiont have to couple their division. In modern cells, the way this happens is that the division of mitochondria happens by pinching from the outside by a host factor called dynamin. Okay, it's a purely mechanical force. It constrains and squeezes until the mitochondrion splits. In bacteria, actually, the way division happens is through an internal machine, which is called FTSZ. But we don't have that internal machine anymore. The host controls it. What we found in our work was actually quite surprising. There are still a few lineages of eukaryotes today which retain the original bacterial division system. So they're sort of living fossils that recapitulate what mitochondria would have looked like several billion years ago, which I find fascinating, right? And these are not model organisms. These are not the kinds, these are not fungi or something, right? They're red algae and certain kinds of amoeba, things that people don't generally study in the lab, which is why they hadn't, this hadn't really been seen in such a manner before our analysis. Okay, I'm going to skip that slide in the interest of time. Okay, so I'm interested in the evolution of eukaryotic architecture, specifically this membrane trafficking system. And what we're trying to do is to explain the series of events that happen from the simple eukaryote to the complex last eukaryotic common ancestor in the absence of fossil evidence. And so the way we decide to approach it is to try and make models of how cells work and then literally do evolution on those models as a way to replay the tape of evolution to see whether there's anything we can learn. Okay, so modeling becomes very important. And so to do this, I have to teach you some cell biology. So here we go, very, very quickly. This is a cell. It's a very, very big cell. Inside the cell, you have a nucleus. Over here, we're only showing one small sector of a cell. This is the plasma membrane. That's the rest of the universe. And what you have then is the nuclear membrane and all these internal compartments. Gray is actually the milieu inside a compartment. So this is a membrane there. It's sort of flattened like a chapati or something. And inside there is the inside of the Golgi, the inside of the endosome, and so on. That's the gray stuff. So all these membranes in the cell are very easily distinguishable because they have different markers. They also exchange material constantly in a manner which I'll describe to you in the next page. And this is very surprising also. When you take a snapshot of the cell in the EM, you just see all this beautiful architecture, but it doesn't give you a sense of the dynamics. Every compartment is exchanging material with every other compartment. Some stuff is going, some stuff is coming, but the composition of the compartment is not changing. So in some sense, the state of the cell is some sort of stable attractor state of a complex dynamical system where there are controls that make sure that the composition of each compartment is maintained over time. This biologists call this homeostasis. There are two types of emergence. The word emergence I'm using in two different ways. One is what I've already described to you, this emergence of the eukaryotic cell over evolutionary time. The second one is the emergence in the mathematical sense. Emergence of this sort of cell scale, several micron scale structure from molecular processes that happen at the protein-protein interaction scale. So that's another kind of emergence. I'm interested in both of them, and they are linked to each other. Okay. One important thing to say is that, as I said, there are different membranes that are decorated with different types of chemicals or different types of proteins. I'll come back to that theme later. What moves stuff between different membranes? It's this object. Okay, it's called a transport vesicle. It's a beautiful thing. It's about 30 or 40 microns in size. It's a lipid vesicle. This sort of dirty green color is the lipid coat of the vesicle. Inside that, there'll be other stuff, but just the surface of a vesicle. This is a reconstruction. It's not real, but just the surface of a vesicle, based on what we know it carries, has all these proteins stuck on it. And this is what moves stuff from one compartment to another. You can think of these as the trucks that move stuff between different warehouses corresponding to different compartments in the cell, the transport vesicle. So how does the vesicle get formed? How does it know where to go? So here's the source compartment. Here's the target compartment. Inside the source compartment, you'll have some soluble cargo and potentially some transmembrane cargo. Cargo, right? Stuff that needs to go somewhere. And so what happens is something called a coat adapter complex comes down on this compartment based on some signal. It makes the curvature to make the vesicle, but it also attracts by protein-protein interactions the correct kind of cargo that have the right affinity and loads those cargo, just the yellow one for example, into that particular vesicle. And once that happens, the coats and the adapters go away and you have the free vesicle. It's floating around and it's got the right cargo. So you've loaded the truck. How does the truck know where to go? If you had an Amazon delivery truck, it knows where to go, not because of what the cargo is, but because he has a delivery manifest and he knows, or GPS ID, and he knows where to go. Vesicles don't have that complexity. Somehow the cargo itself must tell the vesicle where to go, and here's how that works. Somewhere there'll be a target compartment. Remember, there are many different kinds of compartments. One of those target compartments will have one type of protein called a T snare. The vesicle has a protein called a V snare, which is this red guy. And the V snare and the T snare, through specific protein-protein interactions, which, it's based on literally residue level interactions that reduce the free energy, will zip up and use the membrane vesicle to the compartment and deliver the cargo. And the reason this is specific is because different kinds of target compartments have different kinds of snares. In fact, there are dozens of snares encoded in your genome. And one of the themes that emerges from this work is that somehow the availability of many different kinds of markers in the cell allowed the cell to have many different kinds of compartments and many different ways to couple them. So everybody with me so far? You make a vesicle, you load it with cargo. It has this thing called the V snare, which is like an addressing system. It has to exactly couple to the correct T snare because there are many, many options. You can imagine the vesicle diffusing. When it finds the right one, it drops the cargo. Right? It seems inefficient, but it works. Okay. Well, it came from here. Yeah, the red snare is a type of. So in fact, the red snare is a type of cargo that has to come back. Yeah, good. Fine. So this exactly addresses your question. So here's the big picture, right? One thing is that there are different compartments and constantly exchanging matter. So somehow homeostasis has to hold. What goes and what comes has to balance out, even though they're not identical in chemistry. Secondly, the kind of proteins that make budding happen are the same no matter where they are, and you can call them a budding module. The kind of proteins that make fusion happen no matter where they are, you can call them a fusion module. Okay, these modules are the same at the level of sequence, but there are different copies of them. This is called gene duplication. It's a very common theme to drive evolutionary complexity. Somehow gene duplication has increased the complexity of the trafficking system. Duplicate copies of the same modules operate at different locations. If you want homeostasis, every copy of something that moves has to come back in some sort of cycle. And the idea of cycles is highly constraining. And so this is what provides some mathematical structure to the whole problem. Okay, so now I have 20 minutes left, which is good. Here are some of the questions. How did having duplicate gene copies, which I call increased genomic repertoire, drive the increase in vesicle trafficking complexity? I hope I've given you enough cell biological information to motivate that question. To answer it, we need a mathematical framework that has many different levels. It's not simply at one level, right? I want a model that has genes which can duplicate, so you have new versions, or mutate, or get deleted. I want a model that has these modules of proteins that interact. There are specific protein-protein interactions that cause budding and fusion differently at different locations. Somehow through this budding and fusion, I want a network of traffic to form, and this has to be in homeostasis, and you have to have the right flows. And somehow these flows have to be useful to a cell so it can be selected to do some function. So you go from genes to evolution through all these intermediate steps. It's actually very hard to make such a model. It took us a long time to arrive at a particular framework that we now use to describe all this. So I'll tell you our history of doing models of these things over time. I won't spend too much time on it. You can read some of these papers. We started off with just general models of vesicles moving around with random protein interactions to say, can you generate compartments with different compositions in steady state as the fixed points of some sort of distributed dynamical system? And it turns out that you can write down your equations. They have nonlinear terms which correspond to the selectivity of proteins of V snares and T snares, for example. And if these nonlinear terms are sharp enough, you get these fixed points where you have multiple types of compartments, and these compartments are self-sustaining even though they're exchanging material constantly. And in fact, you can have the compartment number, how many different types there are encoded in the protein interaction terms. So this is a type of emergence. We then realized that the topologies that we were getting from these models were too simple. They were basically all-to-all couplings, which is very different from the topology that you get in a cell, which is directed in a certain way. So we moved from, and the problem is that we couldn't explore all possible parameter values for these very, very generic models. We moved to a different framework where we were looking at binary specification of the state space. So you have compartments and vesicles, but now you just say, does it have some substance? Does it not have some substance? Make 0 and 1 models. If it has this composition, it makes this vesicle. If you have this vesicle, it fuses somewhere. You write down those rules. You find the attractor states of the dynamical systems. You tend to find oscillatory fixed points, that is periodic states as attractors. And it turns out that these periodic states are very reminiscent of certain organelles that exist in present-day cells, in particular something called the Golgi apparatus, which is the other half of what my group works on and I'm not talking about today. Finally, we actually did some network motif analysis of the kinds of steady states that arose from these models. You get huge number of steady states, different kinds. But then you find certain motifs that are recurrent, just statistically. And if you look at those motifs, they have certain properties. So it turns out that these properties are absolutely required by certain graph theoretic theorems from the 1970s, like Menger's theorem and Mader's theorem. It has to do with the fact that you need to have recycling and constraints, and it's hard to put a bunch of cycles on a directed graph on an arbitrary graph. And so if you want all the cycles to work out, they have to have edges going in a very specific pattern. So we, in some sense, explain the diversity of systems we had found statistically through the application of graph theory. So I am actually very happy that I'm very proud of this paper, because actually it's stuff that was known since the 1970s, and we had to dig it up somehow. Okay, so I'm going to spend the next 15 minutes talking about our new model, which is going from genes to function all the way through. And this turns out to be, in some sense, at one level, a much simpler model than all the other ones we came up with. So let me walk you through it. So in this model, remember these coat adapter complexes? There are many kinds of coat adapter complexes. You can think of these complexes as the things that determine which cargo will be collected from the source compartment. Okay, there are different kinds. In our cells, there are about 6 or 7 different kinds, depending on how you define it. So imagine a cell that has 3 different kinds of adapters or coats, and I'm coloring them red, green, and blue. Any cargo in the cell will like either just red or red and green or whatever. Like there can be 8 different types of cargo, right? 2 to the 3. There could be cargo that doesn't like to go on any of these trucks. There could be cargo that likes to go on all 3 trucks, right? This cargo, for example, likes to go on the red and the blue, so I'm coloring it magenta. So all cargo is classified into these 8 colors. And once you do that, any compartment or any vesicle can be colored according to what cargo it carries. Yeah, so here, for example, is a compartment that has all the cargo. I'm not showing the black and white, but it has all the others. Here are vesicles. And for example, a green vesicle can only contain these. A green vesicle cannot contain red or magenta cargo, right? Because a green vesicle cannot absorb the magenta or the red. Okay. So, and of course, a vesicle can only get the cargo that's already on the source compartment. So these are the fundamental objects of the model, which are cargo, vesicles, and compartments. The next thing we do is we look at topologies. So we've actually inverted the problem. Instead of creating networks based on protein-protein interactions, I start with the network. I go down the hall and I ask my colleague, a cell biologist, I ask her, what do you think the topology of the cell that you're studying is? And she may say, well, I think there are 3 compartments in the cell, and I think here are the vesicle fluxes that move stuff between the compartments. And so she, gives me a sort of directed graph as a hypothesis for the cell that she's working with. So I take this hypothesis. One thing I didn't mention is that the endoplasmic reticulum, the reason it's so important in biology, all your membrane proteins and lipids are made in the ER. So it breaks the symmetry of the compartment, so I have to take it as a different kind of thing. So the other thing I need to guess is which adapters label which edges, right? So I color the edges. Once I have this as a guess for what the cell is, where the cargo are is just a matter of running the system. It's made in the ER, and for example, magenta cargo will go over there, but it can't come back, and therefore you have magenta here and you have magenta there. So there's a certain regularity to this. It's easy to do this calculation. Once you have this, you can easily guess the colors of all the vesicles and all the compartments on the back of a napkin. Okay, and for example, you'll find things like cargo that go only one way or cargo that go in cycles. You'll find lots of different patterns. Okay, so here we are. We have a network. We have compositions. So this is just one half of the story. What I really want to say is that for some reason, protein-protein interactions make this network what it is. Yeah. So I have to give some explanation for why this vesicle buds from that compartment and why it fuses to this compartment and so on. So here I need to put functional constraints based on what I know about how the biochemistry of vesicle trafficking regulators work. So for example, here's one version of that. For every coat, I must have a recruiter on the surface, and then I must have a V and a T snare that causes fusion. And in this particular model, I also have something called a regulator or an activator of a V snare. I won't get into the reasons for this, but it's necessary. So here are the types of molecules that I need. And then I populate this collection of molecules with colors. So for example, why does the red vesicle come out of this compartment? It's because the red coat likes the cyan recruiter. You see the cyan over there? So that's why red vesicle buds. Then you'll say, aha, but there's cyan over here, so the red should bud also. And I'll say, yes, you're right, there it is. Why does this vesicle fuse over there? It's because the yellow and magenta activator V snare complex likes the red T snare complex, and boom, there's a red there, there's a yellow magenta there, that's why it fuses. And if you spend time looking at this graph, you take a photo of it and go back home and look at it, every one of these arrows has an explanation based on these molecules. So I call this the molecular solution for this network. And for every network, you can write down the molecular solution basically as a long Boolean string. Right? It's a bunch of and or conditions saying what cargo is present and what does it interact with. So roughly, if there are 3 coats and 8 recruiters, there's 24 potential molecules over there. If there are 8 kinds of activators, V snares and T snares, there's 8 to the cubed, 8 cubed molecules over there. So there are, the cell could encode in this very simple model, potentially 536 possible molecules. Whether a cell creates this network can be written as a Boolean function of a 0-1 string of length 536. Okay, I won't tell you how we derive that function, but it's very, very straightforward. And this function is basically written in terms of things that you cannot have, which is anything in the red zone, plus things which you must have, which is I need at least 1, 1 inside each of the blue zones. So you can imagine that Boolean string and how I write it down. Okay, forbidden zones and required zones. Now that we have this, you can take a step back and look at the space of all possible molecular repertoires, right? There are 2 to the 536, crazy number, but 2 to the 536 possible molecular repertoires. And mutation and evolution is walking you through this space. And somehow everywhere you are, there's some network that you're making. And I know the mapping between them because I've written it in terms of these Boolean functions. So for example, the red, blue, and green colors here are not the same as the coats. This is something different. The blue is some particular network, the red is some other network, the green is some other network. Here's an example where 2 types of molecular rules can make the same network. And so over time, the network doesn't change even though the molecules change. Here's an example where the same molecular rule can make both the networks. This is an example of bistability. If you're in that particular molecular rule, you could be blue or you could be red. Here's an example where a single mutational shift can push you from a red network to a green network. Right? So here's my big picture of evolutionary motion through that space. But there's one thing you might have missed. There are some cases where the Boolean expression has no solution. The blue zone is inside the red zone. And so you could ask how many of those there are. So what did we do? We just ran the system. We just searched over all possible topologies up to a certain size. And you can imagine this explodes very, very rapidly because the number of directed graphs of a certain size goes as some ridiculous exponent. We've gone up to 4 compartments and 12 edges, or 3 compartments and 17 edges. So the number of networks, topologies that we've tested is, up to about 4 billion in the 4 compartment system. And the number that have solutions of the molecular type that I've shown is just this number. Okay, so this is one lesson. With random interactions, it's very hard to make a functional and consistent vesicle trafficking network. In retrospect, this is obvious because it's hard to satisfy all these cyclic constraints and all the other things you need to work to make a network, right? So if you just threw a bunch of molecules together at random, you would not get anything. Okay, so that's one lesson over here. So then the question becomes, well, if it's so hard to make a network by chance, potentially is it easier to search by evolution? It's well known that in numerical systems, if you want to guess the solution to a problem, the worst algorithm is to take a random guess and then take another random guess and so on. You always do it by some sort of minimization procedures, right? So let's let evolution do that for us. So to make evolution work, I need some sort of bias in my guessing procedure, right? I need to go from, well, I'm talking about real evolution. Algorithmic, yeah. I'm going to model real evolution on my synthetic cells. So in some sense, it's an evolutionary algorithm. But it's done by duplicating genes, mutating genes, letting them interact. It's done by all the procedures that evolution works by. But I need to bias the process. And one way I'm going to bias the process is the following. I need to somehow, I want to make complex networks. Now, what does complexity mean? It's a very ambiguous term. And I don't really know what function these systems will do for a cell. But we came up with this idea that maybe we could select for systems that are capable of many functions. Systems that are capable of many functions are, in some sense, better suited than systems that are capable of fewer functions. And when I say function, I mean the following thing. Imagine, just as an analogy, that you're living in a city and you have bus stops, and these bus stops don't have any labels. You have no idea which bus stop you're at. You're not going to use a bus stop like that. You're going to use a bus stop if it has a simple and clear label that tells you where you are. So in that sense, if I have a single marker that's only on one compartment, I call that a clear label. And if the more compartments you have with clear labels, the more functionally important that system is. In the same way, you may be at a labeled bus stop, but if your bus doesn't tell you where you're going to go, you're not going to get on the bus, right? What you want is, if you're at this bus stop, I want to know where the bus is going to go based on some marker on the bus. So these are called flow markers, right? And I'm going to say that a flow that doesn't have a unique marker is not really functional because it's hard to use. A flow that does have a good marker is functional because it's easy to use. This idea of easy and hard to use comes from something called affordance theory, which some of you may have learned about from user interface design. Affordance theory means that you should make an object's function obvious just from its design, like having bottle caps that have a specific color, or having a door handle that means pull instead of push. So we're using affordance theory as a proxy for function in these cells. And in this way, for example, here's a system that has 2 compartments and 2 labels. Here's a system that has 3 compartments and 3 different labels. And so this system in some sense is functionally more complex than this one. If you ever get here, you tend not to go back because you get used to this kind of complex lifestyle. Once I add that bias, here's what I can do. I have my networks, remember. Each network has a molecular repertoire that underlies it. In the space of networks, this little dot is this network. That little dot is that network. And I connect them by an edge. If one can become the other can become one. In this case, it's a bidirectional edge. All things that are bidirectionally connected, I make it a little island. You can always go from here to here and back, and the function doesn't change. These are just purely neutral changes. But sometimes you gain new function. Once you go there, you don't come back. So you move from this island to that island. Again, I coarse grain it and I step back. Each one of these dots is one of these islands. And I can go from one island to the other. And this island is, in fact, the set of all networks that have 2 labeled compartments and 1 labeled edge. This one is the set of all networks that have 3 labeled compartments, 1 labeled edge. And so you are increasing in complexity. And here's the full space, the number of labeled edges, the number of labeled compartments. And here's all the sets of networks that can live inside each of them. I'm not showing the whole network. I'm just showing the compartments and edges as you see them. And there's some topological differences between them. So remember what I've done. I've taken tens of thousands of networks and I've sprinkled them into these islands and into these groupings and then into this big space, right? And this is because I have an explicit description of these tens of thousands of networks in my computer. And now I can run evolution, just like I said. Making a network is hard, but maybe discovering it is easy, and that's indeed what we see. If you start off with a proto-eukaryote that has a single compartment, and you just let the evolutionary system run, gene deletion, gene mutation, gene duplication, but whenever you get a more complex system, you keep it. Then what happens is you can make a Markov chain out of this, and you can find n probabilities and so on. And if you do that, then you actually end up accessing, you don't get stuck down here, you end up accessing certain complex states. There are some dead-end states. For example, if you get to this state in this particular model, about 8% of the time, you get stuck over there. There are some dead ends. But a good fraction of the time in this particular model, over 80% of the time, you get to the most complex possible network given just 3 codes. Okay, which is actually very, very interesting. It means that these systems are very hard to make accidentally, but very easy to find evolutionarily. It's a bit like the protein folding problem. There's a funnel that gets you to the final state. This is, has a similar kind of flavor. And one nice thing about this final state, if you look at the topology and you zoom it out, it has all the features that cell biologists love. It has this thing called modified secretion through the Golgi. It has this thing called recycling, which is important for signaling and sensing. It has this thing called phagocytosis, which is important for eating. So even with 3 coats, you can have a eukaryotic system that has all this complexity, which I found very interesting. So you could ask, what's the minimum number of molecules you need to get a certain level of complexity within this framework? Another thing that people spend a lot of time speculating about, imagine a eukaryote that has the nucleus and it has a plasma membrane. At some point in time, it's going to get a new compartment. And by the way, the surface of the nucleus is the ER, if I didn't mention that earlier. So you have the ER, you have the plasma membrane, you get a third. Does the third compartment come from the plasma membrane or from the ER or from somewhere else? And you can actually search through hundreds of thousands of trajectories and see how often you get something from the plasma membrane, how often you get something from the ER, how often you get something from somewhere else, and you can make some statistics out of this. So what we're sitting on now are hundreds of thousands of evolutionary trajectories in the computer, and what we're spending our time doing is looking at reproducible patterns of those trajectories. So this is my last slide. So broadly, what are we doing? I like this sort of framework that I borrowed from this field called assembly theory by Cronin and Walker. So in some sense, there's a universe of possible directed graphs, which are possible vesicular trafficking systems. Some of those are these are just abstract vesicle trafficking systems. Some of those are molecularly possible because they have the right protein-protein interactions. Of those, some of them are historically reachable, right? Over time, there is an evolutionary trajectory that gets you there without killing you at any intermediate step. In biology, this is called contingent. And then you have what's actually there. And so this is a funnel that gets you to this end state. And I think we have a framework to study it at this level. So I'll finish with that. Here's some of the papers on this topic, some of the new ones. Here's my current group. The work I showed you was the PhD work of Sahana and the master's thesis of Kritika. I should just mention for the students, the place I work at, the Simon Center and broadly NCBS, we have a program called the Physics of Life, which is a joint graduate program between physics and biology between NCBS and ICTS, which is a sister institute in Bangalore. You can see that the environment at NCBS really fosters this kind of interdisciplinary interaction. If anybody wants to visit, please come knock on my office door. I'd be very happy to host you, and I'll stop over there. Thank you.